Somewhere in your skull, right now, two types of brain cells are having a conversation. They're directly wired together. One talks, the other listens. No middleman. It's one of the simplest, most textbook circuits in your entire nervous system. A study published this June says that conversation doesn't work the way anyone assumed. Here's the circuit you'd find in any neuroscience textbook. In your cerebellum, the fist-sized structure at the back of your brain that coordinates movement, there are cells called Purkinje cells. Each one sends a direct inhibitory connection to a small cluster of neurons called the deep cerebellar nuclei. One type of cell, one direct wire, one predictable effect. When the Purkinje cell fires more, the nucleus cell should fire less. Simple, direct, textbook. This isn't a fringe idea, it's foundational. Decades of cerebellar research, including how scientists study tremor, ataxia, and dystonia, run on this Purkinje cell, and you can predict what the downstream nucleus is doing. It's the kind of relationship you'd expect from a direct, one-to-one wired connection. Two neurons, one synapse between them. What else would you expect except a predictable, direct effect? In June 2026, researchers in Meike van der Heijden's lab at the Fralin Biomedical Research Institute recorded from both cell types at once, in the same animals, during real behavior, not just in a dish. Their finding? Purkinje cell activity and deep cerebellar nuclei activity did not reliably predict each other, not weakly correlated, not predictable with some noise. The direct wiring diagram did not translate into a direct, predictable relationship in actual, living circuits. This matters because directly wired equals predictable is one of the load-bearing assumptions of systems neuroscience. If two neurons are physically connected by a synapse, we generally assume we can read one to infer the other. That assumption is baked into how researchers interpret abnormal firing patterns in movement disorders. If a Purkinje cell fires abnormally in a tremor patient, the standard inference is, the downstream nucleus is getting a predictably abnormal signal too. What the new recordings suggest instead is that the deep cerebellar nuclei aren't just passively relaying whatever the Purkinje cells send them. They appear to be doing their own independent processing, integrating input from many sources, not just reading out one direct line. The connection is real. The wire exists. But the wire alone doesn't tell you what the receiving end is going to do. Why did the simple version last so long? Partly because it's genuinely elegant. A direct synapse implies a direct effect, and that's an easy story to teach and to build experiments around. Partly because recording both cell types simultaneously in a behaving animal is technically brutal. It's only become feasible with recent advances in multi-site recording. Nobody was being lazy. The tools to check this properly are new. It's worth being direct about what this is. One study, from one lab, published this June. It hasn't been independently replicated yet, unlike the willpower and IQ myths we've covered before, which rested on decades of converging data. This is what a finding looks like on day one, not after 20 years of scrutiny. That doesn't make it wrong. It makes it early. If this replicates, it reframes how movement disorder research reads cerebellar activity. You can't infer the output of the circuit just by watching one input, even a direct one. Researchers studying tremor, ataxia, or dystonia may need to record both sides of the connection, not just the side that's easier to access. And it's a reminder that the wiring diagram and how the circuit actually behaves are two different questions. The textbook circuit. Purkinje cells directly inhibit the deep cerebellar nuclei, so watching one predicts the other. The new finding, recorded simultaneously in real behavior, they don't reliably predict each other at all. The deep cerebellar nuclei seem to be doing their own independent computation, and this is one fresh, unreplicated study, promising, but early. A synapse you can trace on a diagram turned out not to guarantee a predictable relationship in a living brain, and this one's fresh enough that we're watching the story develop in real time. Next time, we're going after one of the most repeated numbers in self-improvement numbers in self-improvement, the claim that it takes exactly 10,000 hours to become an expert at anything. Where that number actually came from is not what you think. Subscribe if you want to be there when it drops.